Hello guys, welcome to the YouTube channel of Gilver Professional Classes. In our previous video lecture, we have discussed about the definitions of mutation and some of its important features. So in today's video lecture, we are going to discuss some of the important types of mutations. So let's start the class. If you consider mutation, the mutation different types, mutation could be of basically two different types. It can be spontaneous mutation or induced mutation. It can be spontaneous mutation or induced mutation. Induced mutation. If you consider the induced mutation is actually caused by some mutagenic agents or mutagens. It is caused by mutagenic agents or mutagens. On the other hand, if you consider spontaneous mutation, they may be due to replication error or due to some spontaneous lesion. Okay, so they may occur due to some replication error or due to some spontaneous lesions. Spontaneous lesions are also there, which are also responsible for the spontaneous mutation. If you consider the spontaneous mutation, they are of different types, basically of two different types. Okay, so you see, if you consider the spontaneous mutation, they are basically of two different types. One type is known as the point mutation, or sometimes it is also known as the substitute mutation. And another type of mutation is known as the frame shift mutation. It is known as frame shift mutation. If you consider the point mutation, the point mutations can be transversion type, it can be transition type. Okay, so it can be of two different types. One is the transversion type, and other one is the transition type. Similarly, if you consider frame shift mutation, there are two reasons of frame shift mutation. Sometimes what happens, one or two nucleotide pairs are changed in a gene. In that case, ultimately frame shift mutation occurs because the reading frame will be shifted. So that change may be due to deletion of one or two pairs of nucleotides from a gene, or due to addition of one or two nucleotide pairs in a gene. So frame shift mutation, it could occur because of deletion of nucleotide pairs or due to addition of nucleotide pairs. So that's why frame shift mutation is of two different types. One is deletion, when it occurs due to deletion of nucleotide pairs. Other one is addition type, when it occurs due to addition of one or two nucleotide pairs. So first we are going to discuss the point mutation. So first you see the point mutation or substitute mutation. Point mutation. If you consider point mutation, it is actually the change in single nucleotide pair of a gene. So point mutation means changes in single nucleotide pair, of a gene. That is known as the point mutation. And one important thing is that if point mutation occurs in a gamete or those cells that are going to form gametes, then those point mutations can be transmitted to the offsprings. That means they can be transmitted to the future generation. So here you write a point. If a point mutation occurs, in a gamete or cells forming a gamete, then it will be transferred to the next generation. Okay, now there is a good example to understand point mutation. and that is the example of sickle cell disease, which what we call the sickle cell anemia. So there is a good example to understand the point mutation. So if you see the example, it is the sickle cell disease. Example, it is the sickle cell disease, or it is the sickle cell anemia. It is the sickle cell anemia. So if you consider this particular disease, this disease occurs due to a mutation, point mutation in the gene that is encoding the beta globin, which is a subunit of hemoglobin. So if you consider, first you see the hemoglobin. If you consider hemoglobin, it is actually made up of alpha and beta subunits. 2 alpha subunits will be there, 2 beta subunits will be there. So like this, suppose this is a hemoglobin, here 2 alpha subunits are there, 2 beta subunits are there. So these 2 are suppose the beta subunits. Okay, in this way it is forming the hemoglobin structure. So if we consider the beta globin is an important constituent. Suppose these are 2 beta, these are beta globin. Along with beta globin, the alpha globin is also there in the hemoglobin. 2 alpha globin, 2 beta globins will be there in the hemoglobin structure. Now if you consider the normal hemoglobin in case of adult, that normal hemoglobin in adult is known as HbA1. This is the normal hemoglobin in adult. This hemoglobin is made up of 2 beta subunits which are normal beta subunits, 2 beta globin, and 2 alpha globins which are also normal, and because of that thing the entire hemoglobin is normal. These 2 are beta and these 2 are alpha subunits. But due to some point mutation, this HbA1 may be converted to HbS, that is ultimately going to form the sickle shaped RBC. Due to this HbS, actually sickle cell RBC is there. Okay, so here you see what happens here. Here the alpha subunit, it is also made up of 4 subunits, but here the thing is that the beta subunits, they undergo certain changes, but alpha subunits are identical. Alpha subunits remain same in both HbS and HbA1. That means HbA1 and HbS, they differ only in their beta subunits, only in their beta globins. So here also 2 beta globins are there and 2 alpha globins are there, but these 2 beta globins are different from these 2 beta globins, and this difference is because of the point mutation. That means single nucleotide is changed in the gene that is encoding the beta globin, and because of that thing entire beta globin is changed, and because of change in beta globin the entire hemoglobin is changed, its entire quaternary structure is changed, and because of change in entire quaternary structure of hemoglobin, this particular hemoglobin is going to form the RBC, which is quite spherical in shape like this. That is the normal shape of RBC. That is the normal RBC shape. But this hemoglobin, this HbS, it is going to form the RBC, which is somewhat like this. It is sickle shaped. It is somewhat like this. It is sickle shaped RBC. This is abnormal shape of RBC. So this abnormal shape of RBC is because of HbS, and this HbS is because of a point mutation in the beta globin. Alpha globin remains untouched. Now how this change occur, you see here. We can understand the thing diagrammatically. Suppose in this particular side, I am writing the condition of wild type beta globin. Here suppose it is the wild type beta globin. And suppose on this side, I am writing the conditions regarding the sickle cell beta globin. So what is the difference you see here? If you consider the wild type beta globin, it is a polypeptide, so definitely it is encoded by gene. Suppose there is a gene, suppose this is the gene, so it is having 2 strands. The upper strand is suppose 3' to 5', so lower strand definitely will be 5' to 3'. So in this particular gene, suppose different codons are there, there is one special codon present, which is suppose the CTC. So it is ultimately there are 3 nucleotides. In the complementary strand, it will be ZAZ. And if this particular gene undergo transcription, it is definitely forming on mRNA. Suppose it is forming the mRNA. This mRNA, if the upper strand is used as a template, then this mRNA will be like this, 5' to 3', and it's the 3 nucleotides written here will form the codon in the mRNA, and this will be the GAZ. So corresponding to these 3 nucleotide pairs, ultimately we are getting the codon in the mRNA. This is the mRNA. After transcription it is formed, and the codon, corresponding codon is ZAZ. and ultimately this mRNA will translate into a protein. Suppose the protein is like this, it is suppose blue colored protein, and in this protein ultimately number of amino acids will be there, but corresponding to this particular codon there is one special protein, there is one special amino acid, that is the glutamic acid, that is suppose glutamic acid, and this glutamic acid, this amino acid, it is corresponding to this particular codon, and this glutamic acid suppose it is present at the 6th position of this particular protein, and this particular protein will be the beta globin. This is the beta globin, and this beta globin is quite normal. Okay. Now what is the case of sickle cell beta globin? So sickle cell beta globin, if you see, suppose it is also encoded by the same gene, so it is 3 prime to 5 prime, it is 5 prime to 3 prime. Here also the same nucleotide is there, but the thing is that here this particular nucleotide pairs, they undergo one change. Here CAC, that means the middle T is replaced by A. The thymine here is replaced by adenine, and because of that thing ultimately in the complementary strand also the 3 nucleotides will be changed. It is now becoming the ZTZ. Now if this particular gene is transcribed, then that is going to form one mRNA. In that mRNA, it is the upper strand is here also, the upper strand is used as a template. So because of that thing ultimately mRNA will be like this, 5 prime to 3 prime, and here the codon will be present. The codon will be changed here because it is now ZUZ. So earlier the codon was ZAZ because the sequence was CTC in the gene, but the sequence here in the gene is CAC, and because of that thing in the template it is CAC, and because of that thing ultimately what is happening, the entire codon is changed and it is now ZUZ. Now this particular mRNA will also form a particular protein, and that protein will also become hemoglobin because this gene is the hemoglobin encoding gene. So that will be hemoglobin. However, this hemoglobin is quite different from the earlier hemoglobin. Why it is different? Because this codon is changed, and corresponding to this codon the amino acid will also be changed. Earlier it was glutamate at 6th position, now it will become valine at 6th position. So at 6th position it will become valine. Okay, and due to that reason ultimately this hemoglobin with this beta globin will be changed, and this beta globin is changed beta globin. So this beta globin is changed, and this is abnormal beta globin with respect to the hemoglobin, this abnormal beta globin, because this beta globin is not going to give you the normal hemoglobin. So this beta globin will give you the hemoglobin that is known as HbS, which is the sickle cell hemoglobin, and this beta globin, this will ultimately give you the normal hemoglobin, that is the HbA1, which is the adult normal hemoglobin. Now why this happening? Why this ultimately this beta globin at the valine at 6th position gives you the HbS, and why the beta globin glutamic acid at 6th position will give you normal hemoglobin? What is the change? Now if you consider the nature of this particular amino acids, if you consider the glutamic acid, it is a polar amino acid. It is polar amino acid. And if you consider valine, it is non-polar amino acid. It is non-polar amino acid. Now, due to this polar amino acid, actually this particular globin is going to form the HbA1. In that HbA1, what we will get? We will get ultimately 2 beta globins like this, and ultimately 2 alpha globins will be present like this. 2 beta globins and 2 alpha globins will be present. These 2 are beta globins. These 2 are the alpha globins. This is the structure of HbA1. Now, these beta globins are having the glutamate at the 6th position. So, due to glutamate, which is ultimately polar amino acid, some negative charges will be developed. One negative charge in this beta due to glutamic acid, and another negative charge on this beta due to glutamic acid. However, if you consider the HbS structure, what is the HbS structure? You see here, it is also made up of 2 beta globin subunits as well as 2 alpha globin subunits. But if you consider 2 alpha globin subunits, but if you consider the structure, these 2 are the beta globin subunits, these 2 are the alpha globin subunits. But these beta globins are different from these beta globins, because in these beta globins at the 6th position glutamic acid is present, due to which negative charge is there, which is polar. But if you consider this beta here, ultimately at the 6th position there is valine present, which is non-polar. So non-polar means it cannot develop any negative charge. So actually in case of HbS, 2 negative charges are different, are less as compared to the HbA1. That means here if you consider in this HbA1, if the number of negative charges n, here in this HBS, number of negative charge will be n minus 2. That means 2 negative charges are less due to presence of valine instead of glutamic acid in the beta globins at the 6th position. So due to that, ultimately this is the polar structure, so it will ultimately, it is comfortable in water, so there is no hydrophobic, it is comfortable in water, it can react with water comfortably, so there is no hydrophobic spot, no hydrophobic spot develop, no hydrophobic spot develop here. But if you consider this particular thing, here the negative charges are not there, it is non-polar, so that's why it will not be comfortable with water, it will not react with water, as a result it will develop hydrophobic spot. So it develops hydrophobic spot, it develops hydrophobic spot, and because of that thing, actually its quaternary structure is different, its quaternary structure is different. However, if you consider the quaternary structure of this HbA1, its quaternary structure is normal, its quaternary structure is normal. But if you consider quaternary structure of this HbS, its quaternary structure is different, and due to difference in quaternary structure, it can change the hemoglobin, entire hemoglobin, and because of that thing, entire hemoglobin change, so it will change the structure of RBC, and here the RBC will become the sickle shaped because of different quaternary structure, and here the quaternary structure of hemoglobin HbA1 is normal, so that's why RBC become the normal spherical shaped. So this is the reason. So here what we have seen, that the sickle cell anemia is because of sickle shaped RBC. Sickle shaped RBC is because of the abnormal hemoglobin HbS, which is developed due to a point mutation. That's why ultimately at the 6th position of beta globin, the glutamic acid is replaced by valine. But in case of the normal case, in case of normal hemoglobin, the HbA1 is there, and in HbA1 at the 6th position glutamate is there, or glutamic acid is there, because there is no mutation, no point mutation in this particular case. So in this way we can see that we can say that the point mutation is because of changes in single nucleotide pair. So this is a very good example of point mutation. Okay, so rest of the types of mutation we will discuss in our next class. So till then, bye bye and take care. And one more thing, if you like the video, please share with your friends and subscribe the channel. Okay, thank you.